So we're going to be looking at the types of transport across cell membranes, and we've learned that the cell membrane controls whether a cell is able to get enough food in, water in, and waste out based upon the size of that cell membrane. So the idea is that simple diffusion is the basic process through which most of this happens, and it requires absolutely no energy by the cell. You've experienced it in a room where maybe somebody does something that is a little bit odorous, and you smell it diffuse from one side of the room to the other. So basically, the molecules are moving from an area of high concentration to an area of low concentration, just like that little video off to the side. Now, the reason that occurs is because of what's called Brownian movement. All molecules, whether a gas, a solid, or a liquid, all vibrate at a certain rate. And because of that, the molecules bump into each other and push each other apart. So imagine being in an elevator with the maximum capacity. All you want to do really is just get out. So it's the idea of moving from an area where there's lots of molecules to an area where there's fewer molecules. So diffusion is a passive process because there's no energy that's being used. So, like I said, those molecules have a natural kinetic energy. So we can see this occurring in liquid when we drop dye into water or food coloring. We see it hit the water and it's in a high concentration, and then as it disperses, we see that the cup of water eventually becomes a lighter color because of the mixture of water and dye molecules. So that diffusion passes through the cell membrane in the same way. So a solute, anything that's dissolved, is going to pass from the area of high concentration to the area of low concentration. All right. So the solute moves down what we call the concentration gradient. Okay? It's kind of like going from a high portion of the hill to the low portion of the hill, if you will. And if you think about it, it's like riding a bike. When you're riding a bike, you don't have to work very hard to go from the top of the hill to the bottom of the hill, right? You can pretty much coast. It's a passive process. Now, osmosis is basically the exact same process, except osmosis is the movement of water across that membrane. So if the membrane is semi-permeable, which cell membranes are, water can pass through, as well as other materials that are small enough to pass through that phospholipid bilayer. So here we've got an example of how a solute, particles might impact the movement of water across the cell membrane. So here we can see on one side I see a lot of green solutes dissolved, and on this side I see very few green solutes dissolved. Now that means which side has more water right now? In the picture, before it even started moving, I see more water on this side and fewer solutes. Because there are more solutes on this side, that means there's less water. So that means that there is a high water potential on one side and a low water potential on the other. The amount of dissolved particles, the amount of solutes that are dissolved, is what affects that. If you think that on each side of that membrane there has to be a 100% solution, think about that, a 100% solution, then that means if the solute concentration is 20%, how much would the water concentration be? If the solution has to equal 100%, that means that the water potential on the left-hand side of that membrane is 80%. Well, what if the salt concentration on this side of the membrane was 50%? Where's there, or how much water is there on this side? Sorry, this drawing or this writing tool is really difficult to use. So if the salt is 50% on this side, ooh, that's really nice looking, Mrs. Learned. See, if it's 50% on that side, how much water is there on that side? Well, if to make 100% solution, that would be 50%. So if there's 80% water on this side, 50% water on this side, that means we get movement of water to the right-hand side of the membrane. So the water molecules use special pores, special water channels. We call these aquaporins. These are special protein pores designed to go through the phospholipid membrane that's to be used only by water, mainly because those phospholipids have that hydrophobic tail. And if the water was exposed to it, then it would disrupt the entire cell membrane. Now, when we look at cells, these are three key vocabulary terms that we're going to use for the remainder of this unit. And they include isotonic, hypotonic, and hypertonic solutions. So do me a favor, go into your note packet and box the words isotonic, hypotonic, and hypertonic. Now, when I look at this yellow cell, I want you to record the amount of salt and the amount of water inside the circle in your note packet. Then in the environment, list the amount of salt and the amount of water. And I want to show you something. A cell in an isotonic solution, okay, that refers to the solution in the environment. That solution in the environment is the exact same as what's in the cell. So when I look at this one, the isotonic solution, iso means that they are equal. So I've put an equal sign in between the cell and the environment. So what is the direction of water movement? Well, in this case, there is no net movement of water. The cell is at what we call equilibrium. So make sure that you record that underneath the cell. The cell is at equilibrium. Nothing's going to happen to it. It's not going to swell up because of an intake of water, and it's not going to shrink. We're always concerned in these situations with what's going to happen to the cell, because we don't want something dangerous to happen to the cells. Now, in this situation, in a hypotonic solution, we've got 10% salt on the outside, meaning we've got 90% water solution. On the inside of the cell, we've got 20% salt. That means we've got an 80% water solution. What's that going to mean for our net movement of cells? So in a hypotonic solution, the prefix hypo means under or lower than. Think of a hypodermic needle. That means under the skin. Okay? The salt solution in the environment is actually less than the salt solution on the inside. So hypotonic is going to refer to those solutes that's dissolved. But what affects the cell is based upon the amount of water. So because there's less salt, that means there's more water. So when I look at the water concentrations, 90% is greater than 80%. And that's what I'm trying to depict there with my little red greater than sign. Because what that does for me is it helps me to see that water is going to go into the cell. So exactly what's going to be the net movement of water? The result is the water is going to move to the inside of the cell. Okay, so you've got one more to look at here. That cell, I'm sorry, I apologize, I forgot. The cell there is going to explode because as water moves in, the cell membrane might not contain it. Think about it. You fill a water balloon too high, probably going to explode. Now, when we look at a hypertonic environment, look at the salt concentrations. Hyper means high, hyperactive, somebody that has high energy, right? So hyperthermia, high body temperature. A hypertonic solution has higher salt concentrations in the environment. Well, what does that mean for the water concentrations? If I use my less than sign, I can see that 85% is less than 95%. So that means, which direction is the water going to go? The water is going to move out of the cell, and the impact on the cell is going to be that it's going to shrink up. Now, we've got a couple of terms to look at and some sample problems at the bottom. So when we look at these cells and solutions, our whole goal is to look at what is the impact on the cell. Will it remain the same? Will it increase in size and swell to the point where it explodes? Or is it going to shrink? So remember, a hypotonic cell, water's going to go into the cell. It's going to swell in volume. If it's hypertonic, water's going to move out. And then if it's isotonic, it'll remain the same. Iso, equal. Don't forget. Now, in an isotonic solution, okay, there's no net movement of water. In a hypotonic solution, there's what's called cytolysis. In a hypertonic solution, there's what's called plasmolysis. Now let's talk about those. So with cytolysis, cells placed in a hypotonic solution will swell and burst. If cells are placed in a high salt concentration, a hypertonic solution, the cells end up shrinking and shriveling. That's called plasmolysis. So cytolysis, bigger, burst. Plasmolysis, smaller, shrink. So when we look at the actual cells, these are real red blood cells that occur. So as we see osmosis, first cell right here is isotonic, here is hypotonic, here is hypertonic. So what happens to blood cells? Well, let's take a look at these and let's see if you can't figure out which ones are correct. See if you can label using the terms isotonic, hypotonic, and hypertonic in each, and then show Mrs. Learned to see if you've gotten them correct.